Every point where a motor nerve terminal physically meets a skeletal muscle fiber creates a highly specialized communication interface called the neuromuscular junction. It is composed of two main structures: the presynaptic motor nerve terminal on the nerve side and the postsynaptic motor end plate on the muscle side. Between them lies the synaptic cleft, a narrow physical gap across which chemical signals must travel. Inside the presynaptic terminal, synaptic vesicles are packed and ready, each one loaded with the neurotransmitter acetylcholine. Each vesicle contains a fixed, discrete amount of acetylcholine. This standardized quantity is called a quantum of neurotransmitter. Transmission does not happen as a continuous flow of chemical, but in discrete, fixed-size packets. The postsynaptic membrane, the motor end plate, is studded with nicotinic cholinergic receptors. These receptors are ligand-gated ion channels, which is a key distinction. They open not in response to voltage changes in the membrane, but specifically in response to acetylcholine binding to them. Without acetylcholine present, these channels remain closed, regardless of the electrical activity happening around them. This makes them pharmacologically distinct from voltage-gated channels found elsewhere on the muscle membrane. And this distinction matters enormously when understanding how diseases and drugs target different components of this system. When an action potential arrives at the presynaptic nerve terminal, the first event is the opening of voltage-gated calcium channels embedded in the presynaptic membrane. Calcium ions flood into the terminal, driven by the concentration gradient. This calcium influx is the direct mechanical trigger, causing synaptic vesicles to fuse with the presynaptic membrane and release their acetylcholine contents into the synaptic cleft through exocytosis. Acetylcholine then diffuses rapidly across the narrow synaptic cleft and binds to the nicotinic receptors on the postsynaptic end plate. Because these receptors are ligand-gated ion channels, binding opens them, allowing sodium ions to flow into the muscle fiber from the extracellular space. This sodium influx depolarizes the motor end plate. That end plate depolarization then spreads to the broader muscle fiber membrane, where it activates a second, distinct set of channels, voltage-gated sodium channels on the muscle surface. These propagate the electrical signal across the entire muscle membrane, ultimately triggering the mechanical events of muscle contraction. To be precise, there are two sequential voltage-gated channel activations during transmission: calcium channels on the presynaptic nerve terminal and sodium channels on the muscle membrane, with the ligand-gated receptor channels on the end plate serving as the chemical bridge between them. Each of these steps is distinct and can be separately targeted by disease, toxins, antibodies, or drugs.